The story started from my point of view in 2007 when GenMab, a small Danish biotech company, had done the preclinical work. From the point of view that myeloma cells express CD38 very abundantly, they had developed a monoclonal antibody that could target myeloma cells. And as you know, myeloma is still a deadly disease, so there was a great need to develop new agents that could target this disease. We know from the lymphoma area that rituximab has made a major difference in outcome for the lymphoma patients. So the hope is that daratumumab, which was the name of this antibody GenMab had developed, would make the same difference for myeloma patients. So the development has been through phase 1, where we carefully dose escalated because we were afraid to see important safety signals, which we did not see. So we dose escalated from very, very low amounts of antibody to up to 24 milligrams per kilo without seeing any problems of safety, except some infusion-related reactions that could easily be managed. After that came expansion studies with larger patient cohorts in phase 2, confirming the safety of daratumumab and showing also a very significant efficacy sign, which is the first time we see clinical efficacy of a monoclonal antibody by itself in myeloma. So there was about a 30% response rate in heavily pretreated refractory myeloma patients by treatment with single agent daratumumab. So this was the platform for starting to think about combinations. And since we believe that daratumumab works by some immune mechanisms involving NK cells, it was very logical to choose lenalidomide as the first partner to combine with, because lenalidomide will boost the immune system, the natural killer cells. So you will have theoretically an ideal partner by combining daratumumab and lenalidomide. So the first combination study was daratumumab with lenalidomide and dexamethasone, and this is what I have reported today. We have previously reported the phase 1 part of it, where we dose escalated dara from 2 up to 16 milligrams per kilo and found no safety signal. So we could proceed with the chosen dose of 16 milligrams per kilo as our new standard. What we see here is from treating 32 patients with the combination of daratumumab, lenalidomide and dexamethasone. We see a high response rate of 81% with a very, very significant high quality response shown by 25% of patients obtaining a stringent CR. This is really, really impressive data when you're treating patients, as is in this case, with a median of 2 prior lines of therapy relapsing with myeloma. So 25% stringent CR, that's really extraordinary. Also, the duration of the response is remarkable. We see rapid responses within 1 month, and we see that the quality of the response improves with time. So the patients get deeper and deeper responses, and they maintain the response for prolonged periods of time. So after a mean observation period of 15.6 months, we have not yet reached the median PFS. So it's about 70% PFS at 18 months of observation. Also a very impressive figure, and an overall survival about 90%. So the good news is that we have an antibody, which means that we have a new agent which has a completely different mode of action compared to what we else have had and seen in the myeloma business. I've heard people earlier today being worried about how to select the most appropriate treatment for their myeloma patients. But what we really have today is something completely different from the others. We have for many years had alkylating agents, and steroids to treat our myeloma patients with. From the mid-2000s, we have had proteasome inhibitors, and we have additions from Velcade to carfilzomib to ixazomib, but they are all the same class of agents, all proteasome inhibitors. We have IMiDs, thalidomide, lenalidomide, pomalidomide, again the same class of agents with some overlapping resistance mechanisms. This is the first time we have something completely different. Totally different mode of action. It kills myeloma cells by interacting with complement, with phagocytes, with natural killer cells, and by itself by binding to the surface of the myeloma cells. And there's also a completely new mode of action revealed. This is presented tonight actually in a poster by Kate Sasser from Janssen and the team from Amsterdam led by Tuna Mutis, showing that there is an immune modulatory effect of daratumumab. So you have in the patients T cells, B cells and monocytes that inhibit the cytotoxic T cells from being active. And these cells are removed by daratumumab so that the cytotoxic T cells can potentially attack the myeloma cells. The take-home message is that we have a new class of drug for treatment of myeloma patients. We have had the alkylators, the steroids, the proteasome inhibitors, the IMiDs. Now we have a new class of drugs that can attack myeloma cells in a completely new way, independent on the resistance mechanisms that may affect any of the other agents that we are using. This is a newcomer.